Hey guys, welcome back. Today we are going to be talking about chapter 21 from Campbell's Biology in Focus, and it will cover the evolution of populations. Okay, so remember that we talked about evolution previously, and we said that for evolution to be occurring, it has to exist in the population. That's actually the smallest unit that can actually undergo evolution. You can't just have one individual that has some sort of weird mutation and suddenly that's considered, an evolved organism. It has to be something that is prevalent enough for the entire population to experience this change, right? Because just evolution is change over time. So one common misconception is that organisms evolve during their lifetimes. That's not true, right? An organism might change over its lifetime, of course, but if it's not the entire population and like a transferring of the gene frequencies over time, it's not true evolution. Evolution takes millions of years. So it's something that you are very unlikely to see in an organism's lifetime. Natural selection acts on individuals, but only the actual populations evolve. Because remember that natural selection, survival of the fittest, only has what's there to work with, right? So if you have an individual that has a specific trait, it's not like natural selection is going to suddenly, endow them with these random traits that just came out of thin air. Natural selection can only act on the individuals with certain traits. It can't give them new traits, right? So consider, for example, a population of medium ground finches on Daphne Major Island. During a drought, large beaked birds were more likely to crack open large seeds to eat them and survive. If you eat, you survive, you're more likely to reproduce and pass that trait on. The finch population evolved by natural selection because the shorter, smaller beaked birds were going to die off because they were not able to eat. That's how natural selection works. Okay, so we're going to talk about microevolution. So microevolution is a change in allele frequencies in a population over generations, right? So this is a long period of time we're talking about. There are three mechanisms that cause allele frequency change. So remember that allele frequency, we're talking about like dominant recessive traits and the prevalence of those traits in a population, right? So the three main mechanisms we're going to talk about today that cause allele frequency change is going to be natural selection, which we just briefly mentioned again, genetic drift, and gene flow. So only natural selection causes adaptive evolution, and we'll get into that in a minute. Okay, so genetic variation is actually what makes evolution possible. Because if everyone in a population, every organism in a population were identical, there wouldn't be any variation to act on. One thing couldn't be better than the other because if everything's the same, then the same trait would be selected for and suddenly you wouldn't have a population if it was a disadvantageous trait. Okay, so variation in the heritable traits is a prerequisite for evolution. So that just means that there has to be a difference in the traits that can be passed on from parent to offspring in order for evolution to even happen. Otherwise, think about if everything was identical, like let's say the coloration or something, and suddenly the environment changed slightly, that now that coloration is disadvantageous and everyone's identical, well then all of the organisms are going to get taken out because suddenly their camouflage is taken away, something like that, right? So Mendel's work on pea plants provided evidence of discrete heritable units that are called genes. So now we know that genes are actually these heritable traits that we're talking about. So genetic variation. So the phenotypic variation often reflects genetic variation. That makes sense. Remember that the phenotype is the way that something looks. Typically, if two organisms look different, they probably have some sort of genetic difference as well. So that equates to genetic variation. So genetic variation among individuals is caused by differences in genes or other DNA sequences. That makes sense because your genes are encoded in your DNA. So some phenotypic differences are due to differences in a single gene or can be classified as an either or basis. So there's just small segments of your DNA that affect the genes. It could be just one gene. It could be a larger string of genes if it's something that's controlled by multiple different alleles. That changes the phenotypic appearance of an organism. And then there's other phenotypic differences that are due to the influence of many genes and vary in gradations along a continuum. So some sort of spectrum where something could fall. Okay, so we have a lot more than just like one specific gene that could cause genetic variation. Oftentimes we have a lot of different genes that are controlling the phenotypic appearance of an organism, and that's where we are going to have a very large source of genetic variation. So genetic variation can be measured at the whole gene level as gene variability. And gene variability can be quantified as the average percent of loci that are heterozygous. So loci is the actual location on the chromosome of the gene. And then you can identify that for the heterozygous individuals. So genetic variation can be measured at the molecular level of DNA as the nucleotide variability. And nucleotide variation rarely results in phenotypic variation. Remember that the nucleotides are like the A, the T, the G, and the C, okay? And remember that we have something called the wobble position in protein synthesis, and our proteins make our structural proteins, so all of the proteins. So sometimes you have different codons that are actually going to code for the same amino acids. So you're not going to have a really big change. But also remember that you have coding and non-coding regions of DNA. You have your introns and your exons. Remember that exons sound like exit, but that's actually the coding region of your DNA. That's the part that you keep, okay? So most differences occur in non-coding regions, introns. Well, that's fine because during mRNA processing, if there's something that happens, some sort of strange nucleotide variation that occurred inside of an intron, that's going to get cut out during mRNA processing, and all the exons are actually stitched together. So you won't even really notice that change all that much while it exists. And then you have variations that occur in the coding regions, which are, remember, the exons. The exons are actually what's kept, and that's what makes your actual mRNA. Before that, it's called pre-mRNA. And the variations that occur in these coding regions, called exons, that you keep, rarely change the amino acid sequence of the encoded protein. And remember that that's because you have that wobble position that's present. The phenotype is a product of inherited genotype and environmental influences. We've talked about how you inherit genes from your parents in our previous lectures. Environmental influences, there's an example of caterpillars coming up in a second. Natural selection can only act on phenotypic variation that has a genetic component. So this is saying that the phenotype can be a product of the inherited genes, but also the environment. And natural selection can't act on the environmental changes. It can only act on the things that can be passed on from one generation to the next generation. That's why it acts on the inherited genotypes. Here's the example of the caterpillars. So these two caterpillars are an example of non-heritable variation. So these are both caterpillars of this particular moth. And they look different, but really their phenotype is different, obviously, because they look different, right? But the difference here is not a genetic difference. It's an environmental difference. So you see in picture A that these caterpillars are raised on oak flowers, and they look like oak flowers. Whereas in picture B, you see that there are caterpillars that are raised on oak leaves, and they look more like the twigs or the branches of this, oak plant that they're on, right? So this is not something that is a genetic difference between these two organisms, while they do have a phenotypic difference. It's based on the chemicals of the food that they're eating in their diets that actually causes a phenotypic change here. So that's an example of our non-heritable variation. Okay, so sources of genetic variation. So how do we get this variety of genetics that that arises with all these different phenotypes, right? So new genes and alleles can actually arise by mutation or gene duplication, which is just the copying of genes. So the formation of new alleles. So a mutation is a change in the nucleotide sequence of DNA. So the A, the T, the G, and the C, it's a change that exists in the nucleotide sequence of our DNA. So only mutations in cells that produce gametes can be passed on to an offspring. So let's think about that. Only mutations in cells that produce gametes. So these are like your oocytes and your spermatocytes that actually go through meiosis to go through the process of creating egg cells and sperm cells, right? They are the ones that are actually passing on their genetic information, right? That your skin cells are not going on to make another human. It's going to be your gametes that go on to make another human. Okay, so it's got to be the germ cells or the gamete cells that are actually affected by the mutation in order for the offspring to get it. Well, that makes sense because the offspring is literally being created from an egg cell and a sperm cell. And if they're not mutated, then there's no problem. But if one of them is carrying a mutation, obviously that mutation will be passed on to the offspring. A point mutation is a change in one base in a gene. And we've talked about point mutations when we talked about protein synthesis previously. So we're going to look at how point mutations can affect the genes. So the effects of a point mutation can vary. You'd think it's a point mutation, it's a change of 1 point. So mutations in the non-coding regions of DNA are often harmless, because if it's the non-coding region of DNA, you're not even going to, see that, because the exons are what's kept. That's what's going to go on and actually create your protein, right? So you won't even really notice that these non-coding regions are being affected at all. And then mutations to genes can be neutral because of the redundancy in the genetic code. And this is part of y'all's protein synthesis project, that that sometimes in that wobble position, you have 3 nucleotides and a codon. That third position oftentimes codes for the same amino acid. If you have a lysine and there's a mutation that causes another lysine, if it's in the same spot, there is no change in that amino acid. The nucleotide structures may change, but it might not always affect the actual amino acid that's being incorporated into that protein, because of the redundancy of the genetic code, where we talked about that with the wobble position in our codons. The effects of point mutations can vary. That mutations that alter the phenotype are often harmful, okay? Because mutations that affect the way that something looks, typically through natural selection, populations are well suited to their environment. And now if you have this phenotypic change, especially if it's like a prey organism that relies on camouflage, and now you have a phenotypic change, it's often harmful to that organism because it was best suited for its environment, and now you throw a mutation into the mix. So is it 100% of the time that it's bad? No, but they can sometimes, if they're altering the phenotype, be harmful to the organism. Mutations that result in a change in protein production can sometimes be beneficial. So you might have a change in the amino acid, but it might not always affect the protein shape and function because of those side groups, the R groups that are on all of those amino acids. Depending on the properties, if they're similar to what it should be, it might actually not change the folding structure all that much of the protein. And then there's sometimes when you have mutant alleles that on rare occasions can actually make an organism better suited to the environment and therefore enhance their reproductive success. And those, of course, are rare occasions, but they're totally possible. Okay, so altering gene number, so the amount, or the position where they're located, called the locus or the loci if it's plural. Chromosomal mutations that delete, disrupt, or rearrange many loci are typically harmful. That makes sense because these chromosomal mutations are not these like small genetic mutations. Like these are on a larger scale. Obviously, if you're deleting, rearranging different genes, you're going to have a typically harmful effect on the organism. The duplication of small pieces of DNA increases the genome size and is usually less harmful overall than like a large deletion or a large rearrangement. Because duplications, if it's something that's supposed to be there and you make too much of it, like typically that's better than not having something at all. Duplicated genes can take on new functions by further mutations. That's also true because you already have one working copy, so perhaps the other working copy could go on to be further mutated that could potentially be beneficial or harmful based on the new function that it develops. An ancestral odor detecting gene has been duplicated many times. In humans, we have about 350 functional copies of this gene, whereas mice have 1,000. And if you think about that, that makes sense that mice would end up with a whole lot more. Like dogs, like the olfactory sensors, they can smell a lot better than humans can. Most other animals can have a more olfactory sensors, so they are better at detecting scents than humans. And if you think about it, like humans are a bit like the exception of evolution in today's standards. Like we are not changing to our environment. We just simply change our environment because that's what people do, right? But like, if it's hot in a certain environment and you have like long, thick, furry, black fur as an organism, you're probably not going to last as long as something that has shorter hair, right? They can't change their surroundings and put in an AC unit in the middle of the jungle, right? So humans today have less functional copies of this particular scent detecting gene, whereas other organisms have high copy numbers of them, which might actually enable them to like detect odors and distinguish different smells that could save them from like toxins or something like that in the environment, or if they got lost and they're able to better identify their own species to get back to where they're supposed to be. That's why, like, cats and dogs are supposed to be able to detect which house is theirs, right? So that's something that has been studied to see that we have a very low functional copy number of this particular gene, whereas other organisms have a much higher prevalence, and that's actually helped them to survive. Rapid reproduction. So mutation rates are low in animals and plants, because if you have a mutation that's really bad, it's obviously going to affect the survival rate of that organism. The rate is about 1 mutation for every 100,000 genes per generation. And the mutation rates are often lower in prokaryotes and higher in viruses. Viruses are something that mutate very rapidly. Short generation times allow mutations to accumulate rapidly in our prokaryotes and our viruses. So if you think about that, you know that prokaryotes divide by binary fission, and they're doing this very frequently, right? Like overnight, if you're trying to grow bacteria on a plate, it goes from something that you can't see to suddenly something that you can see, which is billions of copies of that bacteria. Viruses, the same thing. They're getting into your cells. We've talked about the lytic and lysogenic cycles. We're talking about lytic viral reproduction. It's getting into your cells. It's taking over the machinery. It's making billions of copies of itself very quickly, whereas our cells are not like that, right? So it's more likely that you have a higher mutation rate if you have these higher reproductive rates. So sexual reproduction. So in organisms that reproduce sexually, most genetic variation comes from the recombination of alleles, also crossing over, right? So because you're introducing new alleles together from mom and dad, they each have their own set, right? You're creating new combinations, and then crossing over even further increases the genetic variation of the organisms. So sexual reproduction can shuffle existing alleles into new combinations through 3 different mechanisms: crossing over, which we talked about, independent assortment, which we talked about with Mendelian genetics, and fertilization, just the act of the egg and the sperm cell coming together. Because remember that the egg and the sperm cells in an organism are all going to be different. That's why if you have a bunch of siblings, you are not identical to them, unless you are because you're an identical twin, right? But I have 3 siblings, 14 years older, 12 years older, 10 years older. None of us are identical twins, and we are not identical to each other. That's just the random chance of that one egg meeting up with that one sperm cell to create that individual. That is the process of fertilization. Okay, next we're going to talk about something called Hardy-Weinberg equilibrium. So the Hardy-Weinberg equation can be used to test whether a population is evolving, because all of this is how organisms evolve. This is the evidence. This is Darwin's theory. This is Lamarck's theory. Now we know that evolutions change over time. Now we've learned that it's change over time in a population, and that populations have to have this vast genetic variation in order to be even considered to be evolving, because how is natural selection going to work if there's no variation? So that's everything that we've just talked about. But now Hardy-Weinberg is silly in my opinion, but that's okay. Hardy-Weinberg equation is a mathematical equation. So yes, I'm going to throw some more math at you. I'm actually going to do just a short video of me solving a few of these. So there won't be a whole lot of examples here, but I'll do another one. So it's to prove that a population is evolving, which means it has to fail the Hardy-Weinberg equilibrium, which will make more sense in a minute. So the first step in testing whether evolution is occurring in a population is to clarify what we mean by a population. Okay, because we know only populations evolve. So let's clarify population. Okay, so gene pools and allele frequencies. A population is a localized group of individuals capable of interbreeding and producing fertile offspring. Now I'm going to say that again, fertile offspring. So a lion and a tiger get together and they make a liger. A liger is not a fertile offspring because a liger and another liger cannot have babies. Okay, the same thing is true of like mules. It's a horse and a donkey bred together to make a mule. Okay, they are sterile. They cannot produce fertile offspring. So it has to be individuals who live in the same area that are capable of breeding with each other, producing offspring, and then their offspring can go and produce more offspring. That's what the actual definition of population is. So then a gene pool consists of all of the alleles for all of the loci in that population. So all of the genes and all of the allele variations that are present in the whole population. So an allele for a particular locus is fixed if all the individuals in a population are homozygous for the same allele. Well, that makes sense. You're going to have the same, you have the fixed position if everyone is the same. That makes sense. Okay, so the frequency of an allele in a population can be calculated. And that's kind of what this whole Hardy-Weinberg thing is getting at. Because remember, if the allele frequencies are changing over time, that means that the population is evolving. Let me say that again. If the allele frequencies are changing over time, that means that the population is evolving. Because change over time in a population equals evolution. Okay? So you can actually calculate the frequency of an allele in a population. So we're going to talk about how to do that. So for diploid organisms like we are, okay, the total number of alleles at a locus is the total number of individuals times 2. So let me just remind you that for your genotype for a trait, let's say it's big B, little b, you are heterozygous for that trait. But there are 2 alleles, a big B and a little b. There are 2 alleles. That's why you're multiplying things times 2 here, okay? And that'll make more sense in a minute. So the total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual, and then plus 1 allele for the heterozygous individual. Okay, so for this example here, homozygous dominant, what does that mean? Same, big. So let's say big B, big B. How many times did I just say big B? I said it 2 times. You have 2 dominant alleles, but then you also have 1 dominant allele in the heterozygous, because that would be big B, little b, right? So technically you're dealing with 3 traits there, 3 alleles that are dominant. Are they in the same gene necessarily? Well, for that organism, you have 2 different genotypes for 2 different organisms, but you have a total of 3 different dominant alleles in that situation. The same logic applies for the recessive. If you have like little b, there would be 2, and then your heterozygous would also have a little b in it, so you'd have 3 copies there. Like I said, this is going to make more sense here in just a second. So by convention, if there are 2 alleles at a locus, then P and Q are used to represent their frequencies. Now, there are 2 alleles at any locus. A locus is the location of a gene, okay? So you have big B, little b, little b, little b, or big B, big B. There are 2 alleles at any locus. So the P is representing the dominant, and the Q is representing the recessive. So at any location, you either have PP, PQ, or QQ because it's dominant and recessive. So the frequency of all alleles in a population will add up to 1 because they're talking about a percentage, right? You're talking about how much and what percent is homozygous dominant, what percent is heterozygous, what percent is homozygous recessive. Those things added up will equal 100% for the entire population. So we're just going to simplify that equation, and the standardized equation is P plus Q equals 1. Well, what does that mean? It means P is the frequency of the dominant allele plus the frequency of the recessive allele equals 1. And again, that 1 equals 100%. Okay, so these are the genotypes it's going to use to show you an example here in just a second. So this is obviously going to be incomplete dominance because you have 3 different colors. So up at the top here, you've got your homozygous dominant, you have your homozygous recessive, essentially, and then you have your heterozygous. Of course, these are all dominant traits. That's why it's called incomplete dominant, but this is one variation dominance, the other variation dominance, and then the heterozygous is the blend. Because remember, if it is incomplete dominance, the heterozygous individual is in between the parents. So if you mix red with white, you get pink. Okay, so for example, consider a population of wildflowers that is incompletely dominant, like this, incompletely dominant, okay? So you have 320 red flowers, you have 160 pink flowers, and you have 20 white flowers. So you have a dominant trait, and then both of the dominant traits, okay? So to calculate the number of copies of each allele, you can literally count them here, right? If you have 320 of the C to the R, C to the R, you're going to multiply that by 2 because you have 1, 2, and you have that times 320. So 320 times 2 because you're saying that they are homozygous dominant right here. Why are we adding this number to get the total? Where did this come from? This came from right here, 160 pink flowers. What do you notice about the genotype? Right here, I see a C to the R. Is there only one of them? Yes. So can you double this? No, you just have to use it one time. So 320 times 2 plus this number, which is from the heterozygous individuals, which you'll see here. Okay, so then the same is true for the white flowers, that you have 20 times 2. Why? Because there are 20 of the homozygous. You actually have 40 copies here because you have 20 and 20. And together that makes 40. Okay, plus the heterozygous individuals, because right here you have 1 copy 160 times. So that's how you would get these two totals. So that is P plus Q equals 1. And if you're looking here at percentages, obviously this will add up to be 100%. Obviously here, this actual number is 1,000, but if you put it into percentages, it would be 100%. Here's how they did that. To calculate the frequency of each allele, this was the copies of each allele. No, the copies, how many are in this population. Now we're looking at the frequency. That means percentage of each allele. So the dominant allele frequency, so for the frequency of the dominant red is our 800. Where do we get that? We got that right here for the number of copies divided by the total number of alleles present in that population. How do I know that? Because you have this copy here of the red and this copy here of the white. Do you do copies of pink? No, because you're talking about one allele, which is either C to the R or C to the W. It is one letter, okay? So you're going to divide the total for the one trait by the total number, period, in the population. So you have 800 divided by 1,000 is going to be 0.8, which is 80%. Okay, and then in order to solve for Q, you can rearrange that equation, P plus Q equals 1, to get Q equals 1 minus P. If you know that P is 0.8, 1 minus 0.8 equals 0.2. But you could have also solved that by saying that 200 divided by 1,000 is equal to 20%. Okay, so the sum of the alleles always has to be 1. You double check yourself. 0.8 plus 0.2 equals 1. This is a correct equation. Okay, so Hardy-Weinberg principle describes a population that is not evolving. So the Hardy-Weinberg principle says, if you fit these parameters, this population is not evolving. Okay, all we did here was really figure out the frequency of the alleles. That's it. Okay, but now the Hardy-Weinberg principle is designed to say, hey, is this population evolving or is it not evolving? Because now we know all the things that prove that it is. So let's go ahead and put a mathematical term to it. So if a population does not meet the criteria of Hardy-Weinberg, it can be concluded that the population is evolving. So let's go through that again. If the population does not meet Hardy-Weinberg, the population is evolving. Okay, a lot of people think, oh, if it meets Hardy-Weinberg, it's evolving. It's the opposite of that. If it does not meet Hardy-Weinberg, if it does not agree with what Hardy-Weinberg principle says, then it is evolving. We're using it as like a null hypothesis, sort of. Okay, we're going to go through to actually define Hardy-Weinberg's principle. Okay, so the Hardy-Weinberg principle states that the frequency of alleles and genotypes in the population remains constant from generation to generation. Okay, so what is the definition of evolution? Evolution is change over time in a population. How do we get change over time if they're just like starting to look different? Great, well, that phenotypic variation is typically, not always, typically rooted in genetic variation. If you have genetic variation, then you have a change in the genotypes over time. So Hardy-Weinberg says that the frequency of alleles and genotypes in a population is constant, unchanging, does not change. right? So if we say that no, it is changing, then obviously evolution is occurring. So in a given population where gametes contribute to the next generation randomly, allele frequencies will not change. Again, remember, if you don't meet Hardy-Weinberg, you are evolving. This is where that makes sense. Mendelian inheritance preserves the genetic variation in a population. Okay, so you're still going to have genetic variation, but the causation is a little bit different. Okay, so Hardy-Weinberg equilibrium describes a constant frequency in alleles in a gene pool. So consider that, for example, the same population of 500 wildflowers and 1,000 alleles were this, which we just found previously. Okay, so we have the 80% for the red and the 20% for the white. So if you took those frequencies of those alleles and you made them like little bouncy balls and you put them into a container, 80% of those bouncy balls would be red because that is how frequent that allele is in the population. And the Q, the frequency of the white allele, would be 20%. And again, 80 plus 20 is 100. 100% is correct. P plus Q equals 1. Okay, so if you added a bunch of bouncy balls in 80% for red and 20% for white into this bin, that represents all of the alleles that are present in that population for this particular trait, of course. Okay, so you have the allele frequencies in the population, 80 and 20. And if you put all of the alleles in a large container that represents the overall gene pool for the population, you still have 80% of the possible genes are going to be red, and then you have white as 20. Okay, so if you're assuming that mating is random, which means that there is no selective pressure, it's completely random, that all organisms stand an equally likely chance of reproducing, which we know is not necessarily true, okay? But assuming that mating is random, each time two of the gametes come together, there's an 80% chance that the egg is going to carry the red allele and a 20% chance that it will carry the white allele, because these frequencies did not change. They're just now mixed together in the population, okay? And the same can be true for the sperm. So there is still an 80% chance that that sperm is going to be carrying a red trait and a 20% chance that that sperm is going to be carrying a white trait. Okay, so that's how the gene pool ties into calculating the frequencies in these alleles. Okay, so the frequency of the genotypes can be calculated by saying that if you have 2 red traits, that is p squared. You have 2 of them. You're going to do 0.8 squared. And again, 0.8, we calculated p a long time ago. Okay, so that's what we're going to get for p squared. This p squared represents our homozygous dominant traits. Okay, and then let's do our homozygous quote recessive, even though this is an incomplete dominance question. Okay, our recessive. So you have 2 copies of the white, which you have 20% of getting one of them. So 20 times 20 or 0.2 squared, gives you our homozygous recessive probability, which is going to be 0.04. So these two were easy to find. So we have homozygous dominant, we have homozygous recessive. Let's figure out the heterozygous. It's going to be 2 times P times Q, which means you're going to have 2 times the probability of P and the probability of Q, which will give you 0.32. Okay, so these are the frequencies of the genotypes here. The frequency of the genotypes can be calculated once you get the frequency of the alleles. So the frequency of genotypes can be confirmed using a Punnett square. And I actually cut this off because the Punnett square got really messed up. Sometimes the textbook companies, when they send it to you, it's all gibberish. It's on page 404 in the textbook. Okay. If P and Q represent the relative frequencies of the only 2 possible alleles in a population at a particular locus, then this statement is also true. P squared, which represents our homozygous dominant genotypes, plus 2PQ, which represents the number of heterozygous genotypes, plus Q squared, which equals the homozygous recessive genotypes, will equal 1. Again, it's like a probability thing. The percentage of dominant, the percentage of hetero, and the percentage of recessive will equal to 100% for the entire population. Again, makes sense. So where p squared and q squared represent the frequencies of the homozygous genotypes, dominant, recessive, respectively, and 2pq represents the frequency of the heterozygous genotype. So now we have those two equations, p plus q equals 1, to show the allele frequencies, and then p squared plus 2pq plus q squared equals 1 to represent the genotypic frequencies. Okay, so here's the overall equation here. So the expected frequency of our genotype dominant, the heterozygous, and the recessive. P squared plus 2pq plus q squared equals 1. Okay, so the conditions for Hardy-Weinberg. This is the moment you've been waiting for. Again, the math, simple. I'm going to do a couple practice problems in another video. That way you can sit through and like watch me do 2 or 3 of these. Okay, so Hardy-Weinberg theorem describes the hypothetical population that is not evolving. Again, it describes a population that is not evolving. So in real populations, the allele and genotypic frequencies are absolutely changing over time because we know that evolution is true. But we use the Hardy-Weinberg equation to try to determine if a particular population is evolving. Okay, so the 5 conditions for non-evolving populations are rarely met in nature. Are they never? No, it's rarely. They are occasionally met in nature. Okay, so these are the 5 conditions for Hardy-Weinberg non-evolving populations. So no mutations. This means that the gene pool is modified if mutations alter the alleles or if the genes are deleted and duplicated. Okay, so there are no mutations. Random mating. If individuals tend to mate within a subset of the population, such as their near neighbors or close relatives, like inbreeding, random mixing of gametes does not occur, and genotype frequencies change, right? So random mating says that there is no selection like that. It is absolutely random. Every organism has the same chance of reproducing. No natural selection. So differences in the survival and reproductive success of individuals carrying different genotypes can alter gene or allele frequencies. So this is saying that everything is the same, that you might have, identical genetic variation between all of the organisms, which means that they're all going to be the same. There is nothing for natural selection to actually act on. Extremely large population size. So the smaller the population, the more likely is that the allele frequencies will fluctuate because there's only so few organisms. And then by chance from one generation to the next, that's actually something called genetic drift. So we're assuming that there is just an extremely large population size in order to rule out any minute changes like that. And then we're also assuming that there is no gene flow. So gene flow would be moving alleles into or out of a population, and gene flow can obviously alter the allele or genetic frequencies in the population. So we're assuming that there is none of that. No mutations, random mating, no natural selection, extremely large population size, and no gene flow. Okay, rarely met in nature. These are the conditions for the Hardy-Weinberg equilibrium. Okay, we know that real populations do evolve over time. So this is how you would get a non-evolving population. Okay, so natural populations can evolve at some loci while being in Hardy-Weinberg equilibrium for another loci. So let me explain what that means. So one loci is talking about the location of a gene. Okay, so for some genes, the population can be experiencing evolution, that there's a change in the genetic frequencies for that gene over time. Whereas they are in Hardy-Weinberg equilibrium at a different loci, so for a different gene. So a different gene might not be evolving, but some genes might be evolving, right? Which means that as a whole, then the population is evolving, but it's occurring at like the genetic level because you're looking at the actual frequencies of each of these alleles in a population. So some populations evolve slowly enough that evolution can't really be detected by these mathematical equations. We're going to apply the Hardy-Weinberg principle with PKU, which is a disease in humans. It's called phenylketonuria, PKU, and it's actually a metabolic disorder when you have homozygous recessive alleles, and it occurs in about like 1 in roughly 10,000 babies that are born in the United States. And luckily today they do genetic testing when they're born in order to determine if you have this or not. Because if it's not treated, PKU actually results in mental disability and some other developmental problems. So like when a newborn is born in the hospital, they're tested for PKU like immediately, because you can avoid like the mental disability issues for the most part by just avoiding consuming phenylalanine. Phenylalanine is an amino acid, and that is, it just can't be digested properly in the body with people that have PKU. So you're tested for it, and you can just avoid foods that have high concentrations of phenylalanine in order to avoid most of the symptoms. If you drink like Diet Coke, it has a warning for like PKU individuals should not consume this product. Like if you look at the cans. Okay, so here's the saying that we can assume that the locus that causes PKU is in Hardy-Weinberg equilibrium, given that the PKU gene mutation rate is low and that mate selection is random with respect to whether or not an individual is a carrier for PKU allele. Because remember, it's a homozygous recessive. So if you are homozygous recessive, you know that you have it because you've avoided these foods your entire life, right? If you're dominant, obviously you don't have it. And if you're homozygous or heterozygous, you don't know if you're a carrier or not because you don't have any symptoms unless you go and get tested, right? So mate selection is not based on whether you are a carrier for PKU, because if you're a carrier, you don't know that you're a carrier. Okay, so these are at least the two conditions that are definitely met through the Hardy-Weinberg equilibrium. Natural selection can only act on rare homozygous individuals who do not follow dietary restrictions, because obviously then they would have some sort of mental disability, which is then obvious, right? So natural selection is only going to act on those rare individuals who do not follow the restrictions and then start to develop mental disabilities. The population is large for humans. It's quite large. It's, all people that are occupying a given area. And then migration has no effect because other populations have the same or similar allele frequencies around the world. Okay, so assuming that we meet all of the Hardy-Weinberg equilibrium requirements for this particular trait of having PKU, then we can go ahead and look at the math for it. So the occurrence of PKU is about 1 in 10,000 births. 1 in 10,000 births, you know that PKU is a homozygous recessive genetic disorder. Homozygous recessive is represented by Q squared. Q squared is equal to 1 in 10,000, which is equal to 0.0001 here. And if you square root both sides, you'd find that Q is equal to 0.01. Well, what do we know to be true? P plus Q equals 1. So the frequency of the normal alleles is going to be everybody else. If you rearrange that equation, P equals 1 minus Q, then you really have 1 minus 0.01, which is equals 0.99. So those are individuals that have the normal alleles. And then the frequency of the carriers in this case, remember, you don't know that you have it if you're heterozygous. Heterozygous is equal to 2PQ. Okay, so 2PQ would equal to 2 times the number of the dominant allele, which is going to be 0.99, times the recessive allele frequency, which is 0.01, which gives you 0.0198, or approximately 2% of the US population. Okay, so that's how many people are actually carriers in this case. The frequency of carriers is about 2% of the US population. Okay, so that's a little bit about Hardy-Weinberg equilibrium. We talked about the conditions that need to be met. We looked at the math a little bit. Promise you that as soon as you memorize what P, Q, and 2PQ and Q squared and P squared all equal, it's very simple. The math is very simple, okay? Next, we're going to talk about natural selection, genetic drift, and gene flow, and how they can actually alter the allele frequency in a population. Again, if we are altering the allele frequency in a population, we are absolutely evolving. Okay, so the 3 major factors that alter allele frequencies, so change in allele frequency over time is called, evolution, and bring about most evolutionary change are natural selection, which we've heavily talked about, genetic drift, and gene flow. These two will be a little bit newer for you. Okay, natural selection is the differential success in reproduction results in certain alleles being passed to the next generation in greater proportions. Therefore, if you have a trait that is advantageous, it is a good thing, it will be passed on. And then the next generation will have a higher prevalence of that gene or of that allele, okay? So for example, an allele that confers resistance to DDT, which is like an insecticide, increased in frequency after DDT was used widely in agriculture. So basically we have crops, food that's growing out of the ground on farms, okay? We crop dusted with DDT, which is a chemical to kill bugs. Okay, so a lot of the bugs died. Some of the bugs didn't die, they had resistance. So in the next generation, what do you think happened? The ones that were resistant to DDT have now had babies because they have survived and reproduced. Okay, that is natural selection. So now we have a whole bunch of insects that are resistant to DDT. And actually it caused a bunch of all kinds of crazy things to happen. And I'm pretty sure it's not allowed to be used anymore. Genetic drift. So natural selection is what we've been talking about previously. Genetic drift. The smaller a sample, the more likely it is that chance alone will cause a deviation from a predicted result. So this is kind of like if you have a very small population, very minute changes to that population will have a large impact. Okay, so genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next generation. And genetic drift tends to reduce genetic variation through the losses of alleles, especially in small populations. So these are more chance events, okay? Let's say that your population is a lake, and this lake has fish in it, and it's under, it's in a valley. There's a hill. We don't really have mountains around here, right? But there's a large hill, a rocky hill. And let's say that it experiences some sort of mudslide that causes the rocks to now fall into this relatively small lake because it's a small sample size. That now these rocks are coming in and crushing and killing a large portion of the fish that are in there because it's a small lake, small population, large rocks. That doesn't mean good things for the fish. Now there's only a couple of survivors, and it's not like they were selected for. It was just whichever ones happened to be squished by this chance event. Now that's going to have a huge impact on the allele frequencies in that population because it's actually removing a large portion of the alleles from that gene pool, okay? So here's an example with the flowers again. So in each of these pictures, there's 10 plants. So if in the first picture to the second picture, generation 1 to generation 2, if only 5 of the 10 plants that are present have offspring, or in this case, you have the male and female parts of the flower, they come together, they make a seed, the seed drops into the ground and then grows a new plant, okay? So if only 5 out of the 10 plants have offspring, then your allele frequencies are going to change. So you can see initially for the red, we had 70%, and for the white, we had 30%. And again, that's P plus Q equals 1 was used to determine all of this. In generation 2, we experience a change in allele frequency. For the red, we have 50%, and the Q, we have 50%. Well, that's different from 70 and 30. And then from generation 2 to generation 3, if only 2 of these 10 plants that are present have offspring, you're really only getting the sample size from those 2, right? So then the population for generation 3 is going to have a 100% chance to have red flowers because it happened to be 2 of the homozygous. It shows you with the white boxes, those are the 2 that left offspring. So now generation 3, you have a huge shift from the original generation. But there was no advantageous, this one had seeds and this one did not. It was just a chance event that maybe only these particular plants happened to get pollinated, or the wind carried pollen from this plant to that plant completely randomly. The wind is not selecting for anything, right? That is a pollinator. So you can see in a small sample size of 10 plants, how much, just 2 generations with these random chance events can actually affect the overall population and the frequency of the alleles present. The next thing we're going to talk about is the founder's effect. The founder's effect or founder effect occurs when a few individuals become isolated from a larger population. So I like to be like, okay, this happens on islands pretty frequently. Let's say that there's like a storm and we're dealing with birds because birds can fly, right? So you have a population of birds on a particular island and there's a strong storm happening and there's a bunch of winds and some of the birds that are trying to fly actually get swept to another island. Well, those few individuals have become isolated from the larger population. Right? So now the allele frequencies in the small founder population can be different from those in the larger parent population. Again, this is more by chance, okay? I also like to differentiate this from the other types of genetic variation, like how the alleles change so rapidly, because I like to think about like finders keepers. Like, what does that mean? Like, oh, found new territory, like founder's effect, they're separated. I don't know if that helps you or not, but that's how I remember it. Okay, so again, small populations are going to have a larger change in allele frequency over time. So like if you have a very small section that broke off from the larger population, that allele frequency is going to be very different from the larger group. And then we have the bottleneck effect. The bottleneck effect can result from a drastic reduction in population size due to a sudden environmental change. Okay, this is happening a lot with like ice caps melting and things like that. It's also happened with overhunting, and I'll talk about that in a minute. So by chance, the resulting gene pool may no longer be reflective of the original population's gene pool. Okay, so some drastic reduction in size. Let's say you have 10,000 organisms and suddenly you have 300. That is a drastic reduction in size. Those 300 individuals are not going to have all of the traits that were present in those 10,000 individuals. So you're going to have very different allele frequencies here. And if the population remains small, it may further be affected by genetic drift. Okay, so let's look at an example here. So the bottle is showing you a bottleneck effect. So the original population is all these beads, and the bottleneck event happened. So a drastic reduction in the number of organisms that were in that. So this happened with cheetahs, and it's happened with panthers, cougars, basically big cats in general, because they're overhunted and they are protected, but they are still overhunted. Essentially, if you've killed off a large portion of them, the remaining are going to have to, practice inbreeding and everything like that, which will actually reduce genetic variation over time. But the surviving population is never going to have all of the representation that the original population had. Here you can see that you have a lot of white and a lot of blue and very few yellow. But it looks like the white and the blue are about even, and then a couple yellow sprinkled in. Then you have your bottleneck event, and the resulting population is mostly blue with very one white organism, right? If these are representing the phenotypes, perhaps, of an organism, right? That eventually, a couple generations from here, most of the population will still be this blue trait, right? So that's what's happened to a lot of large cats and everything. So bottlenecking has occurred in wild populations and tends to reduce the genetic variation. In Florida, these panthers were overhunted, and they're actually a subspecies in danger of extinction at this point in time because there's so few of them left. But before they were an endangered species, there was a larger population of them that had larger genetic variation than the surviving population after a bottleneck effect like this. So understanding the bottleneck effect can increase understanding of how human activity affects other species. So the whole thing with global warming, whether you believe in it or not, whatever, there are polar ice caps that are melting, which is obviously going to be removing territory for some organisms and could have an impact on that. Overhunting particular organisms will obviously decrease the number of drastically decrease the number of organisms that are in that particular population, which will affect its allele frequencies over time, which could affect its evolution. I mean, its survivability too. Like a lot of extinct or endangered species experience something like this, and then they just can't recover from it. Okay, so the effects of genetic drift, a summary. So genetic drift is significant in our small populations. Obviously, if you have a huge shift in a small population, you're going to have a much larger impact than if you had hundreds of thousands of individuals in that population. Genetic drift can cause allele frequencies to change at random because genetic drift is a chance event, and genetic drift can lead to a loss of genetic variation within populations, like when we see the bottleneck effect. If it's being removed from the population for death purposes, then those cannot go on and reproduce. If they're not here anymore, they cannot be contributing their genes. And then genetic drift can cause harmful alleles to become fixed, which means that if only a few surviving individuals have these disadvantageous alleles, that now they're going to be perpetuated, even though it's not survival of the fittest. It's just that that's all that's left, right? So genetic drift, again, is our chance events. Our founder's effect and bottleneck effect are examples of this genetic drift. The next thing we're going to talk about is gene flow. So gene flow consists of the movement of alleles among populations. So this is talking about immigration and emigration, so going into or leaving a population. So alleles can be transferred through the movement of fertile individuals or gametes. So for example, pollen. This happens a lot with plants because, like I said, wind and other pollinators pick up little granules of pollen and then fly to other places and go to get more of the nectar from other flowering plants, and they're dropping off little bits of pollen on accident, but they're still pollinating those flowers. So it's very easy for plants to become, experience gene flow because their genes from the pollen, which is really plant sperm, going from one population to another with its pollinators. So gene flow tends to reduce genetic variation among populations over time because the more intermixing of all of these genes that we have between different populations, then you're going to be reducing the genetic variation because you have an exchange of genetic information. So egg cells and sperm cells. So gene flow can decrease the fitness of a population. So for instance, if you have an organism that is immigrating, coming in from a different population that now is introducing a trait into the new population that is not helpful, then it can decrease the fitness of a population. For an example, it talks about the great tit, which is also called Paris major. It's a bird on Dutch islands. The immigration of birds from the mainland, so they're just flying from the mainland to an island, introduces new alleles that are actually decreasing the fitness of the island populations. So natural selection reduces the frequency of these alleles in the eastern populations where immigration from the mainland is low. In the central population, high immigration from the mainland overwhelms the effects of selection. So basically saying that when you have this trait being introduced into the remote areas of these islands, those birds are not being very successful. The offspring of the interbreeding is not being very successful because it's not adapted for that particular island anymore. You're introducing new traits that are not advantageous for that island. So natural selection will eventually reduce the frequency of those alleles over time as those birds die off because they're not being successful. But in areas that you have a very high influx of all these immigrating birds that are coming from the mainland to the closest island, natural selection can't keep up with that. It's just, there's not enough time between the immigration events. That's what that's essentially saying. Gene flow can increase the fitness of a population, on the other hand, if you're introducing an advantageous trait. So consider, for example, the spread of alleles for resistance to insecticides. So insecticides have been used to target mosquitoes that carry West Nile viruses and other diseases. Alleles have evolved in some populations that confer insecticide resistance to these mosquitoes. So the flow of insecticide resistance alleles into a population can cause an increase in the fitness because now they're not going to be, killed off by these insecticides. So for that organism, it is beneficial, but for organisms like, other mammals that live nearby, it's not, because there will be a huge bloom of new mosquitoes. Gene flow is an important agent of evolutionary change in modern human populations. So like I said, humans are kind of the example of the exception of evolution in today's society. But gene flow is an important agent for evolutionary change in our modern populations because you see a lot of, this is especially true for things like skin color and interracial couples that are producing, offspring that have a blend. which is eventually over time going to make the average skin color much darker than it was previously, like centuries ago. Natural selection is the only mechanism that consistently causes adaptive evolution. So evolution by natural selection involves both chance and sorting. So new genetic variations arise by chance, which is our genetic drift side of that, essentially, also mutations, and beneficial alleles are sorted and favored by natural selection. So if you have a new genetic variation, you have these new traits that are, come by chance, that are favored, then they will be perpetuated in the population. Only natural selection consistently results in adaptive evolution, which is an increase in the frequency of alleles that improve fitness. Basically, survival of the fittest. If a trait comes in and it's good, then you will have more of that trait in the next generation and the next generation and the next generation, right? So you're basically increasing the frequency of the alleles that improve your survivability. Okay, so let's take a closer look at that. So natural selection brings out adaptive evolution by acting on the organism's phenotype, the way it looks. So relative fitness. So the phrases struggle for existence and survival of the fittest are misleading as they imply direct competition among individuals. And that's not necessarily true. It's not like, I'm going to fight you over these resources. It's just like, well, if you have a trait that allows you to survive longer than the other one, you're not actually battling for them like a fist fight. You just survive, right? Reproductive success is generally more subtle and depends on a whole lot of factors, actually. So we're talking about relative fitness. It's the contribution an individual makes to the gene pool of the next generation relative to the contributions of other individuals. Okay, so basically, how many offspring are you able to produce to contribute to the next generation compared to the other individuals of the same population? So selection indirectly favors certain genotypes by acting directly on the phenotypes. Again, that's the most obvious thing because you can actually see a phenotypic change, whereas a genotypic change is a lot harder to see. Okay, so these are different types of selection we're going to talk about. There are three main modes of selection of natural selection, like the consequences of it, essentially. So you have directional selection, which is going to favor individuals at one end of the phenotypic range. You have disruptive selection, which is going to favor individuals at both extremes. And there's graphs for this that will really help you understand this. And stabilizing selection, which favors the intermediate variants and acts against the extreme phenotypes. So this is what it looks like. Okay, so you have the original population up at the top, and it's, acting on phenotypes of the fur color of these particular mice. Okay, so these are all the different colors that exist, and the original population has this curve here. And you can see that in each of these images as well. It shows you the original population. And then the dotted line is showing you the evolved population. Okay, yes, the arrows are off a little bit. I told you the pictures don't always transfer very well. Okay, so the original population is the solid. The evolved is going to be the dotted line. So this means that there is a shift to this side. So what did you select against? This is directional selection. Which direction did you travel here? You went for a darker fur color was advantageous. It means that the lighter fur colors were not surviving. Okay, in disruptive selection, where you have the extremes that are selected for, all of the ones in the middle were selected against, which means you have a very low frequency of them. Whereas the extremes were the ones selected for. So in this case, it was advantageous to either be white or very dark, like a dark gray, right? It was advantageous to be one of these two colors in this particular environment. In this environment, you have the original, and now you have just a high prominence of the middle, right? So that is a stabilizing selection because the intermediate, the one in the middle, is the one that is being selected for. Okay, so this just has to do with the environment and what was advantageous in these different environments. You have directional selection, disruptive, and stabilizing selection. And this really look at what that means for the population. Who died out? Why do you think they could have died out? Okay, here, if you had a loss of whatever was like a white background for the mice to camouflage against, then obviously they're going to be seen, they're going to be eaten. Whereas if you were slightly darker in coloration, then maybe you were able to blend in a little bit better, camouflage, and evade predators, right? Just think about different situations in order to help yourself understand these different graphs. Okay, the key role of natural selection and adaptive evolution. So striking adaptations have arisen by natural selection. So for example, certain octopuses can change color rapidly for camouflage. This is obviously going to increase its chance of survival. That is absolutely natural selection. For example, the jaws of snakes allow them to swallow prey that's larger than their heads, which is terrifying, right? But the snake jaw can kind of like unhinge, so it can eat things that are larger than its head. This increases its food sources. So instead of only eating things that are smaller than its head, well, now it can also eat things that are bigger than its head, which if you have more food sources, then you increase your chance of survival. Natural selection increases the frequencies of alleles that enhance survival and reproduction. Makes sense. Survival of the fittest. Biological fitness means that you are able to contribute to the next generation, which means you are having babies. Not, I like to pick things up and put things down. We're not talking about fitness like that. I know it's a quarantine and that's on everyone's mind, but no, we're talking about the ability to have babies and have them survive as well. So adaptive evolution occurs as the match between an organism and its environmental, adaptive evolution occurs as the match between an organism and its environment increases. So it's better suited for its environment. Because the environment can change, adaptive evolution is continuous. It is dynamic. It's always changing, right? And we've said when we talked about natural selection in previous lectures, how important the environment is. If the environment changes, natural selection will now begin to start to select for different traits based on what the new environment is. Okay, so genetic drift and gene flow do not consistently lead to adaptive evolution. So it's not always creating an organism that is best suited to its environment, as they can increase or decrease the match between an organism and its environment. So because genetic drift is a chance event and gene flow is immigration or emigration, these new organisms that are introducing new traits are not necessarily suited for that particular environment. They just happen to get there and procreate. Right? In genetic drift, you're not selecting for the survival of the fittest. It's just a chance event who survived. That doesn't mean that they were the best suited for that environment. Next, we're going to talk about sexual selection. Sexual selection is natural selection for mating success. So it can result in sexual dimorphism, which is when we have a difference in appearance between the sexes in secondary sexual characteristics. A good example is going to be peacocks. So here you can see that the peacocks and the peahens, so that's the male and the female, you have an extreme difference in their phenotypic appearance. That's going to be their sexual dimorphism. There's an intrasexual selection between competing males because they're competing who has the prettiest, brightest feathers that are going to get all the ladies, right? Followed by intersexual selection when the females choose among the showiest and most extravagant of the males. So that's what I was just talking about here. So intrasexual selection is competition among individuals of one sex. Typically, the males are competing for mates of the opposite sex. So the males are competing for the attention of the females. Intersexual selection is often called mate choice, and it occurs when individuals of one sex, typically the females, are choosy in selecting their mates. They know that it's more, quote, expensive for them to commit to that relationship because they only have so many eggs that are going to get fertilized and all of that. They're going to have to go through the whole gestation period, deliver those eggs, or in this case, we're talking about birds, right? Or for humans, you're going to go through like 40 weeks of pregnancy, which is actually like 10 months, not really 9 months, to have a whole human child come out of your body, right? So usually the choosier mate is the one that has a more expensive cost at procreating, okay? That's typically how that works. A male showiness due to mate choice can increase a male's chance for attracting a female while decreasing his chance of survival. Think about that. These peacocks with these beautiful tails, let's say that it needs to suddenly run away from a predator. Well, its tail is going to get it all the ladies. or its tail is going to get it some tail if you catch my drift. But it's also going to slow it way the heck down, because if a predator is coming, you think that is going to be able to move very fast. Look at how many feathers there are there. Okay, it's going to be a lot harder for this thing to get away from a predator. So it's actually decreasing its chance of survival, even though it is increasing its chance of passing on its traits, which is kind of crazy how all of that works. So how do female preferences evolve? So the, quote, good genes hypothesis basically suggests that if you have a trait that is related to male genetic quality or the health of that male, then hopefully your offspring would have the same result, right? Both the male trait and the female preference for that trait should increase in frequency. So these big, showy, beautiful, brightly colored tails are considered to be good genes among peacocks and peahens, right? So the females are going to go for the males that have the longest, brightest, showiest tails. They will both have an increase in the frequency. Okay, so the preservation of genetic variation. We're going to talk about something called neutral variation. So neutral variation is genetic variation that does not confer a selective advantage or disadvantage. It's just a change. It doesn't mean that it increases or decreases a chance of survival or fitness. Various mechanisms help to preserve genetic variation in a population. So there's something called diploidy or diploidy, however you would like to say it. So diploidy maintains the genetic variation in the form of hidden recessive alleles, obviously through the heterozygous individual. So heterozygotes can carry the recessive alleles that are hidden from the effects of selection. You know that in a lot of cases where recessive alleles, if you have a double recessive, so a homozygous recessive individual, typically like that means that they're the ones with the disease or whatever. But if you are the heterozygous individual, you do not have the disease. You're in fact a carrier in a lot of cases, but you might get a benefit. It's called the heterozygote advantage. And that's essentially what this is saying here. We'll talk about, I think it gives you an example of all of that in a minute. You also have something called balancing selection, which occurs when natural selection maintains stable frequencies of two or more phenotypic forms in a population. So balancing selection includes the heterozygote advantage and the frequency dependent selection. So we're going to get into those next. So the heterozygote advantage that I was talking about is basically when the heterozygous individual has a higher fitness than either of the homozygotes. So they're dominant or recessive, that the heterozygous individual has the highest chance of survival. Natural selection will tend to maintain two or more alleles at that particular locus. And then, for example, the sickle cell allele causes the mutation that in the hemoglobin that also confers malaria resistance. So basically, sickle cell anemia causes a mutation in hemoglobin. So it changes your red blood cells to create this sickled kind of like half moon shape or like a crescent moon shape, which has, negative effects of being able to carry oxygen correctly around the body. It also can, I believe it can also cause a higher frequency or a higher likelihood of having blood clots because your cells can kind of fit together more like puzzle pieces than bounce off of each other like round cells should. But individuals that are heterozygotes for sickle cell anemia, they have some sickled cells, but also normal cells. So they have enough normal cells in order to provide enough oxygen to all the tissues of their body, but they're also resistant to malaria because of that. So you can see that this is like a little map to show you the frequency of the sickle cell allele, and it's very frequent in certain areas where you would have malaria, right? So you'd have a higher frequency of like the heterozygotes for the heterozygote advantage in villages and cities and things that are near areas that are ridden with these mosquitoes that carry Plasmodium falciparum in this case, which is going to be the intracellular parasite malaria. Okay, next is the frequency-dependent selection, which occurs when the fitness of a phenotype declines if it becomes too common in the population. So frequency-dependent, if it's too common, it starts to decline, right? Fitness starts to decline. Selection can favor whichever phenotype is less common in the population, so it's not necessarily one or the other, it's just whichever is less common. in a particular area. So, for example, frequency-dependent selection selects for approximately equal numbers of right-mouthed or left-mouthed scale-eating fish. So there's these particular fish, and the mouths are kind of like veering towards one side or the other, whether it's the left or the right. And they are selected just frequency-dependent, whether it's one or the other, equally likelihood, but whichever one is less common is going to be the one that is favorable, because their prey is going to start to realize what's going on, right? So if you have a high prevalence of all the right mouth eating fish, then they're going to be watching, their prey is going to be watching out for them. And now suddenly you have another one that's eating from the opposite side, like a sneak attack. It's less common until it becomes more common, and then it would flip again. That's kind of how frequency-dependent selection, it's a little finicky how it works. This is an example of this fish I was talking about, the veering mouths, right? You'd have a kind of like a back and forth as to which is more common. So why natural selection cannot fashion the perfect organism? So you don't have the perfect organism because the environment is changing. There's things around the world that are constantly changing. So selection can only act on existing variations. You can't just introduce the new genes that are going to make an organism perfectly suited for its environment, or a population that is perfectly suited for its environment. Evolution is limited by historical constraints as well, and adaptations are often compromised because it might be really good for catching a mate, but then also not very great for your survival, things like that. Chance, natural selection, and the environmental interactions between an organism and where it's actually living, those are all going to be out of your control, right? Especially chance of these genetic drift events, and also gene flow events. You can't control any of that. You're always going to have variation. And sometimes it might be for the better or the worse, just depending on the size of the population, which event is actually occurring in that population. And natural selection just tries to keep up as best it can with creating organisms that are strong and fit for that environment. But is it perfect? No, because you can't keep everything. You're not living in a lab where the conditions could be perfect all the time, right? You're living in the real world where things change all the time. Weather can have a huge impact. Like I said, with the genetic drift, hunting, things like that. The environment is changing. The organisms kind of have to change and adapt with that. So that's why you can't actually create the perfect organism. But natural selection does a pretty good job at keeping organisms very well suited to their environment. Okay, that is it for this lecture. We are going to finish up with chapter 22 next. And that is it. That is it for this class. So thanks for sticking with me on this one. We have one more chapter that's going to finish up our evolution unit. It's going to be the origin of species. And that'll be it for us. So you guys have a great day. Stay safe during your quarantine, and I'll see you next time.